Hello, everyone. Welcome back to Chapter 2 of Cognitive Psychology, Psych 4100. In this lecture, we're going to be discussing cognitive neuroscience. So let's remember that cognitive neuroscience is sort of the middle ground between cognitive psychology and neuroscience. So in this chapter, we're going to talk a little bit about the brain, some of the brain's major structures, some of the sort of finer grained details about how the brain communicates with itself, as well as some of the larger order ideas and theories about sort of more global brain connectivity and brain activity and how those things produce functions, right? Behaviors and thoughts, which are the domain of cognitive psychology. So a few things to think about that we're going to maybe touch on, or that the things we touch on might prompt some of these questions. What is cognitive neuroscience? Why does it matter? How does the brain transmit information from one place to another? How are things in the environment, such as people's faces or trees, represented in the brain, right? This idea of representation is going to come back because your brain cannot literally store a picture of your mother's face in itself, right? There is no file drawer for photographs in your brain. So somehow your brain has to remember what your mom's face looks like without literally being able to refer to a picture of her face. In other words, your brain has to represent your mother's face somehow within itself, and it's using its activity. And then lastly, what are neural networks, these sort of large global issues of connectivity throughout the brain, and how do they determine cognition? So first and foremost, a definition of cognitive neuroscience. Cognitive neuroscience is the study of the physiological basis of cognition. So if a cognitive psychologist is interested in a type of thought or behavior, a cognitive neuroscientist is interested in how the brain produces that thought or behavior. So this involves an understanding of both the nervous system as well as the smaller subunits of the brain and the nervous system that comprise this system. So, for example, you can think about the brain as one big sort of global thing, one big organ like your heart or your liver. But in reality, the brain is divided into these subparts that are divided mostly along physical and functional sort of parameters. So, for example, the occipital lobe back here at the back is associated with the visual system. The parietal lobe is associated with movement and the perception of movement. The temporal lobe is associated often with things like language. And the frontal lobe is involved in what we might call higher order decision-making, strategy, personality, things of that nature. And then within your brain, sort of internal to your this sort of shell that we call the cortex that's on the outside of the brain, we have a whole nother system known as the limbic system. We'll go through all this stuff in a little more detail later. But the point here is to keep in mind that, yes, your brain is one big organ, but we can also divide your brain into these subsystems that each have very specific functions. But then these subsystems also interact with one another. They talk to each other. They send information back and forth to one another in order to produce human behavior. So one thing we need to sort of get out of the way before we move on into the complexities of cognitive neuroscience is the idea that we can vary our level of analysis when we talk about cognitive neuroscience, meaning it is not more or less correct to focus on a single element of the brain or of behavior. So something like, let's look at A over here, right at the top right, this picture of a man looking at a woman, right? And he perceives this woman, right? What we would call, he sees her. His brain is registering her face, the landscape back behind her, how close she is, how far away she is, right? What has she done to her hair? Your brain is registering all of this input to produce a perception. And this perception is what exists in your conscious mind. Right? It's the act of your engagement with her in the world. However, we could break down that perception at many different levels of analysis. We could ask what groups of brain structures communicate with each other in order to produce this perception. We could ask, are there specific individual brain structures that are activated? We could ask questions about individual neurons that are activated. And then furthermore, within those neurons, we could ask questions about the very basic biochemical processes that produce neuronal activity, right? Because if we want to explain how and why this man perceives this woman's face, we could answer that question as, well, there are some sodium ions that move into and out of a neuron, and that produces electrical activity. That's a valid level of analysis. But it also needs to be sort of propped up with or rounded out with these higher levels of analysis that also provide other levels of explanation. So we can explain his perception at any single one of these levels of analysis, but we actually are better off attempting to explain his perception at all of these levels of analysis. Each viewpoint adds a small amount of information, and when we really put all of those viewpoints together, we have a much more rich understanding of what creates his perception. Right? So you might have a cognitive neuroscientist who looks more like a biologist who's studying just very basic chemical processes. And you might have a cognitive neuroscientist that is looking at global brain activity, but they're essentially all part of the same process of trying to understand this sort of basic rudiments of how the brain produces human behavior. So we can't talk about the nervous system unless we talk about its building blocks. Right? What is the nervous system made of? It's made of neurons. There's other stuff in there, but the main functional component of the nervous system is neurons. These are cells that are specialized to create, receive, and transmit information. Each neuron is made up of three major parts. We have the cell body right here where the nucleus is contained of the neuron. The neuron is no different than any other cell in your body. It has a nucleus. Then the neuron has this axon, this sort of tail that pulls away from the cell body. And this axon ends in what we call axon terminals. And then connected to the cell body are what we call dendrites. And these dendrites is where the neuron receives information. Think about dendrites. I like to think of them as a game of hungry, hungry hippos. So each of these dendrites is a hippo, and what it's doing is it's reaching out into the space in your nervous system, and it's gobbling up information. That information comes in the form of neurotransmitters. You've heard of these things before, like serotonin or dopamine. So these dendrites are gobbling up neurotransmitters. And once they collect enough neurotransmitters, they produce a signal that comes down and collects at this place where the axon connects to the cell body. We call this the axon hillock, Hillock. The axon hillock. So all of these neurotransmitters that the dendrites are gobbling up, you can think of them as sort of producing this effect where we get all this energy that collects right here. And then once that energy builds up enough, boom, we get an action potential we get the propagation of an electrical signal down the axon, just like an electrical signal moves through the wire that charges your computer. It's actually probably pretty similar. It's a good analogy. You get this electricity moving down this wire, this axon, and when that electricity hits these axon terminals are, think of them as balloons that pop. And when those balloons pop, they release their own neurotransmitters. And then the next neuron over here to the right somewhere, that's not that we don't see right here in this image, there's another neuron over here, or another few neurons that are gobbling up those neurotransmitters with their own dendrites and then continuing to propagate the signal. So dendrites gobble up neurotransmitters. They send that signal to the axon hillock. The axon hillock builds up all this energy, and then when it gets to be too much, the gate opens, an electrical signal comes down the axon. That electrical signal pops the balloons that are the axon terminals, and those balloons release their own neurotransmitters, and then they talk to the next neuron. So communication within a neuron is electrical, but communication between neurons is actually chemical. So we have two types of communication happening in the nervous system: electrical communication within a neuron and chemical communication between neurons. These yellow things that we see here are called the myelin sheath. They're basically insulation for the axon. So again, the analogy of charging your computer comes back to be helpful. The cord that charges your computer from the wall outlet is wrapped in rubber, right? That's an insulator. And so you don't actually see the wires, right? If you were to cut your cord, I don't recommend it, but if you were to cut your cord, on the inside of it, you would see all the wires that actually transmit the electricity. But wrapped around those wires is rubber and plastic so that those wires are insulated and can transmit the electricity more quickly. That's exactly what myelin does. Myelin simply accelerates the transmission of this electrical activity and makes neurons more efficient. Interestingly, myelin is made of fat cells, like lipids, which is one of the reasons why it's very important for young children to eat lots of fat content, whether through breast milk or through formula, because if they don't have enough fat content in their diet, then their brain has trouble making this myelin, and then it impacts their cognitive development substantially. So this is basically what I said before. The cell body contains the nucleus, which is going to keep the neuron alive. The dendrites are these branches that reach out and receive information from other neurons. These are hungry hippos. And then the axon is this tube filled with fluid that transmits the electrical signal down to the axon terminals that release their own neurotransmitters. So we can see here at the top, this is what your average neuron looks like in the cortex, basically in your brain. We've got this sort of spiny tree-like spreading of dendrites and a similar but usually smaller spreading of axon terminals. Now, neurons that are receptors, meaning these are the neurons that actually measure information from the world, look a little differently because you have a stimulus coming from the environment, and you have in the cell body of this neuron, you have a specialized receptor that measures some kind of quantity from the world. So in this case, if this is a touch receptor, then it might measure pressure. And so the more pressure that you put, for example, on the tip of your finger, the more quickly this neuron fires and it gets everything started. Now, when this sensory neuron fires, it is in direct contact with the world, but then everything beyond this, once the signal is released to the next neuron, then every other neuron in the chain that leads all the way up to your brain is simply interpreting the input from this single neuron. So sensory neurons are the only neurons that actually measure the outside world. All other neurons in the nervous system in the cortex are simply measuring signals from other neurons. Now, when we were first learning about the brain and neurons, the original theory was that the brain was basically this net of neurons called a nerve net, similar to a highway system where information or cars was just sort of nonstop flowing and continuously communicating throughout this singular network. But then these two Spanish physiologists and neurophysiologists named Ramon y Cajal did some pioneering studies on the physiology of neurons, and they founded what's known as the Neuron Doctrine. And what they said, and what is now true, we know to be true, and is the case for neuroscience, is that the appropriate level of analysis for understanding communication in the nervous system is not a singular nerve net, but rather that individual nerve cells are transmitting signals, and they're not necessarily continuously linked to all other cells. We have networks of cells that fire together, and it's a sequence of individual neurons that communicate each with each other, not necessarily this one large net that's constantly communicating with itself. So really what's happening, and what you're seeing here, I like the picture on the right a little better because you can see a single neuron. Here we see an individual neuron. This is a cell body. We've got some axons, and if you could zoom in even further, you'd see some dendrites probably right here, as well as some axon terminals. These individual cells are the ones doing the communicating, and those individual cells build up into what we're going to call functional networks. But those functional networks exist to perform individual functions. They don't exist as a singular net of communication. They are dedicated structural and functional connections that over time sort of grow together and build together in order to produce very specific functions. So we really want to think about the functioning of the brain as starting at the level of the neuron, so the neuron doctrine, and then building up from there, rather than starting at the level of a singular nerve net. So the electrical signal within a neuron is called an action potential. That's the burst of electrical activity that comes from the axon hillock and goes down to the axon terminals. So this happens when neurons receive information from the environment, whether that is the outside environment in the case of a sensory neuron, or whether it's other neurons in the case of cortical neurons. And we can measure those action potentials with little microelectrodes that can pick up the electrical signal. And it looks like this. So neurons are at rest, meaning nothing's happening, right? There's no propagation of an electrical signal, and they actually have a slightly negative electrical charge. And then if we get this buildup of information at the axon hillock, boom, the gates open and this impulse is driven down the axon. And what we see is that the electrical signal becomes positive. We get this spike of positive electrical activity. And this propagates itself all the way down the axon and then returns back to normal at its resting state and will fire again. if enough information builds back up. So neurons are basically sitting there firing, 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 firing. Think about them as producing like a spark, right? A spark, 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 spark. And that's how they communicate. The quantity of electrical activity never changes. So the movement from a negative 70 millivolt charge to a positive 40 millivolt charge will remain consistent across the life of a neuron. What changes is the frequency with which a neuron fires. And we'll talk about that later when we talk about representation, because that's the major tool that neurons have to produce representations. Is not the magnitude of their electrical activity. The magnitude stays the same. What changes is the frequency of firing. So, like I said, size isn't measured. It remains constant, but the rate of firing changes. So, for example, if you were just to caress your arm, so take one of your hands and just sort of caress your forearm, that's a very low intensity stimulus. And what's happening is your sensory neurons are firing slowly. They're going boom, 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 boom. Versus a high intensity stimulus, if you were to slap your arm very hard, then what happens is your neurons register that high intensity stimulus and transmit it to your brain and tell your brain that, hey, this one is different, because they fire quickly. They go boom, 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 boom. And that's how your brain tells the difference between a caress and a slap. Slow firing indicates a caress. Fast firing indicates a slap. And we can take that analogy and we can use it for all of your senses. Right? It works like that, more or less that way, in a sense, for all of your senses. We can tell the type or intensity of a stimulus based upon the frequency of neuronal firing, as well as maybe the location of the neurons that are firing. Again, A is what maybe a light pressure on your skin would look like if each of these lines is the firing of a sensory neuron. B would be a moderate pressure. C would be the firing of neurons to strong pressure. Now, when neurons, excuse me, when neurons communicate with each other, they don't actually touch. They come unbelievably close to touching, but there is a little bit of space between them. We call that the synapse. It's the space between the axon terminals of one neuron and the dendrite or cell body of the next neuron. And so this little space is where neurotransmitters exist. That's where the hungry hippos of the dendrites gobble up all the neurotransmitter. They explode into this space, and then the next neuron has to gobble them up. And in fact, many psychotropic drugs and medications work because they act on that synaptic space. They manipulate the amount of neurotransmitter in that space. So, for example, a common class of medication used to treat depression are SSRIs, selective serotonin reuptake inhibitors. Basically, those medications work because they force more serotonin into the synapse than would typically be there. And that boost in serotonin often helps in the relief of depressive symptoms. So this is a depiction of a synapse. What you're looking at over here in the sort of zoomed in circle on the right is the synaptic space between a axon terminal. These little nodes are the neurotransmitter that is getting forced into the synaptic space. And down here, we have the dendrites of the next neuron gobbling up all of that neurotransmitter. And in fact, these little spaces, what we call receptors that gobble up the neurotransmitter, they're basically like locks, a series of like door locks. And neurotransmitters are chemicals that are the keys to these locks. So some of these locks can only be opened by serotonin. Some of these locks can only be opened by dopamine. Some of these locks can only be opened by epinephrine. And in fact, the reason why things like illicit drugs work so well is because illicit drugs often mimic particular neurotransmitters and thus kind of trick these receptors into thinking that they're neurotransmitters. So your brain naturally produces opiates. Your brain uses them to help relieve pain in case you get hurt. If you were to take heroin, which I do not recommend in any way, shape, or form, if you were to take heroin, you are essentially injecting a synthetic opiate into your system that tricks your nervous system into thinking that it's a endogenous, is what we call it, an internal opioid that your body produces naturally. You're sort of replacing it with a synthetic man-made opioid that unlocks these receptors and provides the sort of feeling that you get if you were to take opiates. Okay, so now that we know a little bit about how neurons work, we're interested in sort of bridging the gap between this biology of neurons and the mind, right? Cognitive psychology. So somehow your brain has to, and your mind has to, represent the world using this biological system. So without that representation, we would be unable to act in the world. And so a large part of cognitive neuroscience is studying this principle of neural representation, because everything that you experience, every thought, every feeling, everything you hear, everything you see, everything you think, everything you remember is due to the functioning of neurons. Nothing else produces cognition. That's it. It's all due to the basic biological functioning of these cells. Which begs a question: how the hell do these little dots of neurotransmitters equal the memory of your mother's face? And that's an incredibly deep, often philosophical, as well as biological and cognitive question to answer. And we have some pretty good theories and evidence about how your brain represents the natural world around it. So one of the basic ways that your brain does this is through what we call feature detectors. This original research was done in the 1960s, and yes, it was actually done with cats. Hubel and Weissel dropped recording electrodes into the visual cortex of cats, and then they showed cats various geographic shapes that were moving in different directions. So they might show them sort of a tilted rectangle to the right, then they might show them a tilted rectangle to the left, then they might make this rectangle move. Then they might show them a smaller rectangle that moves in the other direction. Just all these different combinations of geographical figures and movements. And what they found is that there are particular neurons that respond to particular features of the environment. So in your visual cortex, you literally have neurons that all they do is fire when you see a rectangle. Then you have other neurons that all they do is fire when something moves up and to the left. Then you have yet other neurons that all they do is fire to the color green. You have other neurons that all they do is fire to triangles, so on and so forth. We call these neurons feature detectors because their job is to detect particular specific features in the environment. And so what your brain can do is your brain can say, okay, the green neurons are firing, the triangle neurons are firing, and the up and to the left neurons are firing. Thus, what we're seeing is a green triangle moving up and to the left. And so your brain is capable of representing that object and that movement based upon the firing of a combination of specific neurons. Now, this is to some extent inborn, but it is also dependent upon experience. We know that the structure of the brain changes with experience. We call this plasticity. And in these original experiments, Hubel and Weissel examined this plasticity by taking away experience from particular kittens. So they would take kittens at birth and basically put these little goggles on the kittens where the goggles only provided vertical stimuli for the entire lifespan of the kitten. And so, not surprisingly, when the kitten grew up, it could only perceive vertical stimuli. It was incapable of perceiving the horizontal elements of normal stimuli. So, while your brain is prepared to produce these feature detectors and use them to represent physical stimuli in the world, your brain literally needs the input in order to figure it out, right? Your brain has to go through a process of sort of culling through objects in the world and culling through your neuronal responses so that it can produce adult-like behavior and perception. So these feature detectors are the lowest level of what we call hierarchical processing. If you take any object in the world, you can break that object down into its pieces and parts, right? Its color, its shape, its movement. You have think of a pyramid. At the bottom, at the base of that pyramid, are feature detectors. And then as we move up the pyramid, we get more hierarchically complex neuronal detectors, meaning that as we move forward in processing in the brain, you start to get neurons that don't respond to the shape of a triangle, but rather they respond to the shape of a human body, which is much more complex, but is its own sort of object. And then we also get neurons that fire to faces. They don't fire to features, they fire to a face, or they fire to bodily movements, for example. So we have you could think of a hierarchy of detectors. At the bottom of that hierarchy are feature detectors, color, shape, movement. And then as we move up the hierarchy, the detectors begin to detect increasingly complex types of stimuli. Now, this begins to happen through different patterns of responding, right? So remember our discussion of levels of analysis earlier. We can't just explain the brain's activity and its functioning at one level of analysis. We have to build upon levels of analysis. So specificity coding is what we call the activity of feature detectors. It's the representation of a specific stimulus by the firing of specifically tuned neurons that are specialized for that stimulus and only that stimulus. But then as things in the world get more complex, as our perceptions get more complex, we move into what's known as population coding, which means that there might be a pattern of multiple neurons that all fire together to represent a particular stimulus. So you quite literally have, for example, in specificity coding, particular neurons that only fire when you see a face. But then you have a population of neurons, maybe, I don't know, 10, 30, 50, 1,000 neurons, that their job is to fire together when you see your mother's face. And then you have a different population of neurons that fire together when you see your significant other's face. Then you have a different population of neurons that fire together when you see your sibling's face. So we can move from single neurons that code for or represent a specific stimulus, and we can then move to populations of neurons that synchronously fire together to represent a particular stimulus. And then we can even have the lack of firing of neurons to represent a particular stimulus, or sparse coding, meaning your brain can represent a stimulus by the firing of just a small group of neurons with other neurons remaining silent. So this is what it might look like. In specificity coding, neuron number 4 might be responding to Bill, neuron 9 responds to Mary, neuron 6 responds to Raphael. If we looked at a larger network of population coding, then these 10 neurons might fire in this particular pattern only to Bill. These 10 neurons might fire in this particular pattern only to Mary. These 10 neurons might fire in a particular pattern only to Raphael. And the same is true for sparse coding. So this is how your brain represents the outside world. It uses a combination of specificity coding, population coding, and sparse coding to determine what is in the world around it. A not terrible analogy is the ones and zeros that we use to build computer coding. So think about, and again, the analogy is not perfect, but it's not a bad analogy. Specificity coding. If we were to code for Bill, it would be 00010000000. If we were to code for Mary, it would be 00000000010. Right? Got it? And then we can carry that analogy forward to population and sparse coding. So maybe Bill is 01110000100, whereas Mary is 10001111011, right? Something like that. So it's not exactly how it works, obviously, but it's a nice analogy to help sort of understand what we mean by coding. And you can think about it in terms of computer coding with ones and zeros. Okay. So this is not a bad place to stop about halfway through this lecture. The next part of the lecture, we're going to dive into more the structure of the brain and what those major structures do, their functions, what they're associated with. And then at the end of the next part, we're going to discuss some more theories about more global issues of connectivity throughout the brain, in which these major parts of the brain might be talking to each other, rather than what we've been talking about in the first half of this lecture, which was individual neurons talking to each other. So The first half of this lecture, we started with individual neurons and how neurons talk to each other, how they build to maybe population coding. And then in the second half of this lecture, we're going to talk about major brain structures and then how those major brain structures might talk and pass information to one another to produce global connectivity and behavior. So thanks, everybody. Check out part 2 and finish up chapter 2, cognitive neuroscience, as soon as possible. Thanks, everybody, and I will talk to you later.